I am Professor Bhatnagar. I retired as a senior professor and head and dean from Department of Zoology, College of Science, Mohanlal Sukhana University. I am specializing in cell and molecular biology and taught to postgraduate students cell and molecular biology as well as animal biotechnology for the last 39 years. Today we are going to talk about the gene and its manipulation. But before we talk about the gene manipulation, let us understand what is DNA. In this first slide, you can see there are few diagrams. The above diagram on the left side is actually a bacterial cell, and bacterial cell is the single cell structure, and you can see here that there are certain strand-like structures. One is circular and another one is spread in the green area. The first one, which is red, is basically the DNA or a chromosome of a bacteria, and you can see that there is outside nothing. They are freely in the bacterial structure. The another one is circular, and they are basically called as plasmids, but definitely they are also DNA. But this is also known as a special DNA present in bacteria. This type of structure is not present in plant or animal cells. Now, the other two diagrams are basically, the upper small diagram is basically the virus, and there also you can see the rolling structure in the center, which is also a DNA. And that is a DNA of a virus. Virus consists of both DNA and another nucleic acid, which is known as a RNA, ribonucleic acid. The lower two pictures are basically the animal cell structures, and the first one is basically the nucleus, which is always present in the center of a plant or an animal cell. All cells have the nucleus because this is basically the structure which is controlling the activity of a cell. In the center of a nucleus, then again there is a small dot. You can see there, this is called as a nucleolus, which is basically responsible for controlling the activity of a nucleus. And then you can see the fibrous structures spread throughout the nucleus. These are basically the chromosomes. In the normal condition, or in the resting phase, which we call, before the division of a cell, the chromosomes appear like a thread. And you can see here, these large number of threads are interwoven in the nucleus. But what is chromosome? Now here you can see the structure shown as inside the cell. There is a cell, then there is a nucleus, then you can see a 4-strand structure, which is basically a chromosome, and that chromosome is basically made up of a DNA. The entire chromosome structure is basically nothing but DNA and certain proteins, which are basically outside the DNA. What is DNA? The structure of a DNA was first time shown by Watson and Crick in 1952. And the Wilkins also suggested the basic structure of a DNA. So all the three were given the Nobel Prize for suggesting the structure of a DNA. If you see the basic structure of a DNA, it consists of 2 strands, and they are basically coiled along an invisible axis. You can see small striations in between the DNA coiling. These are called nucleotides. There are about 10 nucleotides between one complete turn of a 2 DNA strands. Okay? If you see these nucleotides in detail, you can see that the first nucleotide here consists of T and A. So T stands for thymine and A stands for adenine. They are basically nitrogenous bases. They are structures which consist of basically nitrogen. In the next nucleotide, if you see, there is written A and T, so adenine and thymine. Then in the third, you can see C and G. C stands for cytosine and G stands for guanine. So basically in the formation of a DNA, there are 4 nitrogenous bases. One is thymine, adenine, guanine and cytosine. The 2 nitrogenous bases in one nucleotide from both sides, or we can say that the thymine as well as the adenine connected with sugars. In DNA, the sugar is called deoxyribose. Towards thymine, if you see, the sugar is always directed upwards, while in the case of a adenine, the thymine is directed towards downwards. Similarly, in the second nucleotide, adenine is again connected with the sugar deoxyribose and T is connected with the deoxyribose. In between, so these are basically the two nucleotides. In between these two, there is a structure called P, a circular dot consists of a structure P. So P stands for phosphate. So basically, the two nucleotides are joined together with a phosphate group. A complete nucleotide is a phosphate group, deoxyribose sugar, nitrogenous base like thymine connected to adenine, adenine again connected to deoxyribose sugar and then connected to phosphate. In fact, the DNA is basically labeled like this. When the oxygen which is present in a deoxyribose or O is directing towards upside, that side is known as 3' and the lowest nucleotide, if you see where there is a OH structure is there, then we call it as a 5'. And on the other side, because the reverse condition is there, so 3' is at the base and 5' as the top. So here, if a very important phenomenon, the DNA of one strand goes from top to bottom and the another strand coiled on this strand in reverse direction. Okay. So this is basically a structure of a DNA. And the details of this can be seen in this diagram. In this diagram, I have included another structure, single-stranded thread-like structure. This is basically called as RNA, ribonucleic acid. So RNA is always a single-stranded, while DNA is always double-stranded. And RNA is basically synthesized over DNA by certain specific enzymes. RNA has large number of important roles in our cell. Okay. Now, what is gene? And what is difference between a gene and a DNA? DNA is a long structure. If you spread a DNA, coiled DNA, it is said by scientists that even you can, if you round around the sun or around the moon, I think about 10 to 50 turns you can make. So in a single cell, such a large DNA is present. But gene is a small unit of a DNA. DNA may consist of 1 lakh nucleotides, but the gene consists of only 200 nucleotides or approximately 200 nucleotides. So, and gene is very specific because it has some functions. It has to do some functions. In such a large DNA, there are certain areas. One is known as an intron, and then there is a structure called exon. So if you spread a DNA and if you try to watch this DNA structure, you can recognize intron, then maybe again intron, then an exon, then intron, maybe exon, then again intron, then again exon. Means intron is basically the part of a DNA which is not functional, which is not active. But in the case of a exon, they are basically active. And they produce basically responsible for different type of activities of a DNA, which I will discuss later. Now, what is engineering DNA? First thing, let us talk about a DNA replication. Well, DNA replication means basically formation of a new DNA strand on the existing DNA strand or a parental DNA strand. So, if you have single DNA structure, a new synthesis can take place on this parental structure. How? Actually, for replication, we are not going into details, but basically the two strands get separated to certain area with the help of certain enzymes. Once they are separated, the first on one of these two strands, the synthesis can occur, but sometimes the simultaneous synthesis in both the structures are also observed. This actually happens at the time of cell division, means when the cell undergo division to form two cells. See, in every nucleus, whether it is a plant or an animal or a bacteria, a definite amount of a DNA is present. But see, if a cell undergoes division and when it forms two, so naturally the nucleus also start dividing. when the cell is undergoing division. So first nucleus division undergo division. So the DNA will become half. But as we say that for every species there is a specific DNA content in the nucleus, in that case, if a normal DNA content is divided into two, then the amount of DNA will be less in the two newly formed cells. And if the amount of DNA is less, the characteristic of that species should change, because the content required for that species character is not present. That amount is always required for that. So in that case, before the division, cell starts preparing and it does replication. So new DNA synthesis takes place just before the division. New synthesized DNA accumulate in the cell, and then ultimately cell undergo division. Some scientists say that for the division also the excess DNA synthesis is required, and when the amount of a DNA enhanced, then the normal amount of DNA required, the cell undergo division. So first DNA manipulation or the gene engineering is basically related with the DNA synthesis or DNA replication. Sometimes DNA undergoes a break. At a certain point along its long length, on one of the strands of a DNA, a small break takes place. That break is possible because a break in between the two nucleotides or at one nucleotide. And you can see clearly in the diagram here, which is on the left side. This break in a DNA strand is called as nick. And very often it occurs. But DNA has the ability to repair it, and that phenomena is known as actually a DNA repair. We are not going into details of this, but this is one of the phenomena related to DNA engineering. The another phenomena is called as a DNA methylation. Very important phenomena. Several times it happens. Even in the laboratory test or laboratory activities, when we want to modify the DNA structure or we want to silence the structure of DNA, the DNA methylation is done. DNA methylation means adding a CH3 group on a nitrogenous base. It actually, you can see that at a C group in a nitrogenous base, CH3 is placed. CH3 is a methyl group. So H group or OH group which is present on the CH is basically removed and CH3 insert itself. Here in the next diagram, you can see a small DNA structure, and on the upper side you can see 2 methyl groups added, and on the lower side you can see 3 methyl group added. These addition of a methyl group on a DNA changes its characteristic. Scientists do it as a manipulation also to change the activity of a DNA. I think you might be aware with the name of Dr. Hargobind Khurana, an US-based Indian scientist of Indian origin. In 1970, Professor Hargobind Khurana was succeeded in synthesizing a gene in a test tube in the laboratory by using different types of nucleotides, sugars, and phosphate group. And with the specific enzymes to synthesize DNA, he synthesized a DNA. Considering this fact, he was actually given the Nobel Prize in 1970. I'm not going into details of this. Certain more important things are there in relation to the gene engineering or gene manipulation. How gene manipulation is possible? See, if you want to do the gene manipulation in a laboratory, you have to remove the DNA from a cell. So what we do? Here you can see in this diagram, which is a step 1. There are 4 test tubes. You can see in the first test tube, the tissue, suppose a small piece of a tissue taken from a liver, it is completely minced in a mortar and pestle and then diluted in a specific solution, and then it is treated with specific enzymes. So the extra things which are present in this solution, like some proteins around the DNA or certain specific enzymes or any other substance which is present here which can interfere in the later reactions, they are actually destroyed. And then finally, in the last tube, the DNA is basically present, which is very cautiously removed from the test tube and can be used further. Here you can see a diagram This is basically showing the DNA structure. If you have DNA samples and if you want to find out what kind of DNA it is, what kind of genes are present, what kind of nucleotides are present, and if you want to identify the complete sequence of a DNA, that in what sequence the T, A, G and C are present in the DNA, in that case this particular technique is very important, known as gel electrophoresis. So what we do, that DNA which is extracted in the earlier step, basically run over a gel plate. There is a small glass plate over which a gel substance is spreaded, and small holes are prepared on the top of it. Then electricity is provided. The DNA which is extracted is placed in these small holes, and if you run the electricity, pass the electricity on this gel, in that case the DNA runs from top to bottom. While running, the larger part of a DNA remain on the upper side, and which are smaller parts goes to the downwards. Sometimes what people do is, since the DNA is very long, they break down the DNA, and after breaking, with the help of an enzyme, they run it like this. So small DNA goes at the bottom, the large DNA present on the top, and this way the different fragments of DNA are separated. This is also a very important technique which is used in a DNA manipulation. Another technique is known as a PCR. The full form is polymerase chain reaction. Very important technique. Can you believe it? This technique has revolutionized the cellular and molecular biology. PCR technique is basically used to amplify the DNA. What do we mean by amplify? Amplify means from a single DNA strand or from a single DNA fragment of a strand, we can produce large number of DNA, a large amount of a DNA within minutes. It is said in the cell and molecular biology that this is the single technique which has completely revolutionized the cell and molecular biology, because earlier the particular technique of amplifying DNA used to take 6 or 8 months. Now you can do with the help of a small equipment called polymerase chain reaction or thermal cycler. You can do this technique within 3 to 4 minutes, and large amount of a DNA can be produced. The technique is basically based on a high temperature, like a 90 degree, where the DNA breakdown. Then immediately the temperature is lower down, and a specific enzyme is added which works on this particular condition, and that basically synthesizes the DNA. So large amount of DNA can be, a cycle goes on, high temperature, low temperature, high temperature, low temperature, and ultimately you can see in these two panels that a large amount of the DNA can multiply from 2 to 4, 4 to 8, 8 to 16, 16 to 32, something like this. And within seconds or within minutes, you can produce large amount of DNA. Now, once the DNA is produced, now you want to identify a particular gene sequence in a DNA. So large number of fragments are separated. Now you have to separate them. This technique here is also related to the gene manipulation and known as a Southern blotting. And it was actually identified or developed by a person called Southern. That's why it is called as Southern blotting. In the first panel, the same electrophoresis is done. On the top, you can see the small holes. These fragments of a DNA are placed in these holes, and then you can see in the first panel, the DNA is separated. Now, question is how to identify it. So what we do normally, we transfer this on a paper towel. A small paper, specific paper, cellophane paper is placed on the gel after the run of a DNA. So this DNA present on the gel actually binds to a paper towel, and then actually this paper, which is a nylon membrane, you can call it, is basically treated with a specific type of things. Like if you want to identify a particular DNA sequence, then a probe is required. Probe means the already synthesized DNA sequence on which we want to work. When we add the probe on this paper towel, in that case that probe binds to the DNA structure, and then you know the sequence of this particular probe, and that's why you can identify the DNA sequence of the DNA strand on which this probe binds. So this is another important technique, Southern blotting, required to identify the structures. Next step is basically the cloning of a DNA. Cloning means basically introducing a foreign DNA into a host DNA. Let us understand what is host DNA and what is foreign DNA. Basically, a host DNA means we have a DNA structure or DNA identified or isolated from a nucleus. Now we want to add a small fragment of a DNA taken from another species or from another animal. As I earlier said, that scientists have tried to introduce the characteristics of a tomato, like a red color and its taste, into potatoes. How you can do it? This is possible only because by our cloning. So what we do in this, if we take a small amount of a DNA or a gene responsible for a specific characteristic or responsible for a red color of a tomato and want to transfer into potato, in that case, the host DNA will be potato and the foreign DNA will be tomato's gene. So this tomato gene will be introduced into a potato DNA. Now how it is possible? It's very easily done in the laboratories. And for this, the circular DNA of a microorganism is required, or the bacteria is required. Almost all bacteria consist of these structures. These are known as plasmids, and they are also called as vectors. Vectors means which are transport vehicles. So the foreign DNA, small foreign DNA, is basically transported to host DNA with the help of this circular DNA or plasmid DNA or vector. Okay? In the diagram, you can see here three different types of natural plasmids or vectors. I need to explain before I go for small details of a cloning so that you can understand it, the phenomenon completely. See, this first structure basically comes from a bacteria called Escherichia coli. Do you know that Escherichia coli is present in your intestine? It is a helpful bacteria present in our intestine even from the time of our birth. It consists of a small circular DNA which is basically known as which is basically known as a plasmid E. coli, or it is a circular structure and it consists of various parts, such as you can see there is a written ORI, O-R-I. ORI means basically origin of replication. This is the part which should be present if the large number of copies of this plasmid structure is to be prepared. Another is taken, or it is not a natural structure, but it is basically artificial structure developed in a laboratory using the E. coli vector or a plasmid, and by adding certain other characteristics. This is called as a PUC19. P stands for plasmid and UC stands for University of California, where it is basically developed. And number is it's the variation, means from 1, 2, 3, 4, 5 up to 12, 13, 14 and 19th variation, and that's why it is called as a PUC 19. Well, certain new things are added to it. Normally in a plasmid, there are certain structures, like one is called a selectable marker. Two selectable markers are present. One is for ampicillin, another is for tetracycline. Both are antibiotic. If they are present in a plasmid, they produce resistance to the bacteria. If you place a bacteria in a plate where these antibiotics are present, but because these resistance structures are present in them, or the part present in them, the bacteria will not die because of this. But if you remove these from the bacteria, then the bacteria will die in the presence of ampicillin or tetracycline. So they are basically used as a marker to understand the addition of a foreign DNA into the plasmid. Okay. So in the second structure PUC19, you can see clearly there is a AMPR, which is basically a AMP or ampicillin resistance gene present in the plasmid. There is another gene which is known as a tetracycline resistance gene, which is also present. The ORI is always required because multiple copies has to be produced. But another important thing which is present here, which you can see here on the yellow area on both the sides of a blue gene structure. These are basically known as the restriction sites. Restriction sites means there is a specific enzyme, because when you want to introduce a new DNA into the vehicle or into the vector or into the plasmid, in that case you have to first break down it. So to break it, a special enzyme is used known as a restriction enzyme. Till now, about 3000 enzymes are identified from the different bacteria, which can basically break down the plasmid structure. How we can do it, or what is basically a restriction enzyme? Let us take an example of bacteria E. coli or Escherichia coli, which is present in our intestine. This bacteria consists of the plasmid structure, but in addition to that, two specific enzymes are identified. One is known as a E. coli or EcoRI. R stands for the restriction and 1 stands for the first enzyme. Similarly, there is a EcoR2. These are the two enzymes which are restriction enzymes. And they always identify or break down a specific sequence, which is a 6 nucleotide sequence on a DNA. And this 6 nucleotide sequence is exactly like an English word madam, M-A-D-A-M. If you read from the left side, it is same, madam. If you read from the right side, again it's same, mada. So if on a DNA, if this kind of a sequence is present, in that case, these restriction enzymes act on it. And surprisingly, on our DNA or in different animals' DNA, plant DNA, if you see the sequence of a DNA, you will find this kind of sites or these madam kind of restriction sites present on the DNA. But these sites are always present on the two sides of a particular gene structure or exon. So it is easy to cut because there is one restriction enzyme identify these both the areas, and therefore it will cut it and remove the DNA. Another enzyme which helps in addition of a DNA, known as a DNA ligase. So, and in the third is, if you see the most complicated structure known as a PBR332, and again you can see EcoRI, Hind 3, EcoRV2, BMHI and SALT. They are all basically different kind of restriction enzymes which are present on the structure. How it is possible? Well, see in this diagram very clearly. I have shown the double-stranded DNA sequence. So you can see CCCGA, and then in red I have shown the madam restriction site. GAA TTC on one strand, and on the opposite strand CTTAAG. As I had told you earlier when I was showing the structure of a DNA or describing the structure of DNA, I told you G always pairs with C and T always pairs with A. So GAA, and in front of that CTT, and then in the upper you can see TTC, and then on the lower side AAG. So if you read from the left side, on the first strand GAA TTC, and on the second strand, if you read from the right side, GAA TTC. So the same reading, and that's why it's similar to them, the word madam. This particular site is basically in our subject known as palindromic sequence. These kind of sequences are many, and well, this is a 6 nucleotide sequence, but there are also sequences which are 4 nucleotide, 3 nucleotide, something like this, which are present on the DNA structure as well as on the plasmids. So these restriction enzymes which can identify this kind of a sequence can be used to cut the DNA. Since DNA, this particular enzyme cut on this kind of sequence only, that's why it is known as a restriction enzyme. If we have this kind of a structure and DNA is cut, then you can see that in the lowest structure where I have shown two lines and I have written staggered cuts. They overlap each other, but some part is left which is not overlapping. These type of cuts are basically called as staggered cuts, and they are very important for joining the new DNA or the two DNAs. See, if you have two papers, if you directly try to join two papers by their what you call edge, you cannot join. But if you overlap the papers, you can easily join two papers. The same thing is here. So an extra nucleotide sequence is extended on both the strands, and that helps to join the DNA. But sometimes, as I told you, here is a 6 nucleotide sequence, but there are also certain cases where the 4 nucleotide sequence is there. So if you remove the 4 nucleotide sequence, in that particular case, what will happen? There will not be a structure like this, and there will be a structure known as a blunt ends, as I shown in the slide on the topmost line. Now, if there is a blunt end, as I said, the two papers cannot be joined with at the edge. So similarly, the blunt ends, if you try to join the two DNA, it will not be easy to join these together. So what normally we do, if we have the blunt ends, in that case we extend the ends of a first nucleotide sequence or a DNA strand of a upper side on the left side, and we extend certain nucleotides or add certain nucleotides on the lower DNA on the right side. So we, in the laboratory, we try to make them as a palindromic sequence, which can be used further for the phenomena. Now, how the cloning is done? The cloning is done in this diagram. It's very clearly explained. Circular structure is a plasmid, and the linear structure is a genomic DNA. Genomic DNA means a DNA which is present in the nucleus of a particular cell. The entire DNA present in one cell nucleus is basically called as a genomic DNA. Now, this genomic DNA, if you are interested in a particular sequence, it is with the help of a actually restriction enzyme, maybe EcoRI, we have to cut this DNA. So DNA will cut at various places where the palindromic sequences are present. Small fragments are formed. Similarly, the plasmid will be also cut. In the sequence, you can see that the plasmid is cut and there are small fragments of a DNA. Now, one of this DNA will be added to the plasmid. So in the next step, you can see that the small DNA fragment is added and the DNA circle is complete, or a plasmid circle is complete. This is possible with the help of an enzyme called as a DNA ligase. Once the new DNA is added to a plasmid, as it consists of a DNA fragment from another DNA, which is basically a foreign DNA, then it is known as recombinant structure. And since it is a plasmid, we call it as a recombinant plasmid. Since by this technique we can produce a recombinant plasmid or recombinant structure or recombinant DNA, the technology is also now known as recombinant DNA technology. Now, once this takes place, in this diagram certain details are there. Now, you have produced the DNA, recombinant DNA, which is present here in the figure. But in the next diagram, you can see a bacteria. Now, you require to multiply these structures. Because see, if you use a single plasmid structure in which the foreign DNA is introduced, can you manipulate it easily? It's so small that you cannot see from the naked eye. And it will remain in a solution. You may lose it. So you require a large amount of DNA so that you can easily manipulate further. In that case, it has to be multiplied. And what is the easiest method to multiply? Well, we can transfer this recombinant plasmid into its host DNA. Means it can transfer into, since we initially took the plasmid from an E. coli bacteria, now introduce into an E. coli bacteria. And this plasmid has a natural capacity to multiply and produce large number of circular DNA. So large amount of circular DNA is produced. Now you can break down the bacterial DNA and circular DNA can be removed. This will be again breakdown, and then it can be transported or can be introduced into any cell wherever you want in a different way. These are the basic techniques of a DNA manipulation. When the DNA is basically used in a DNA manipulation, there are three important terms are used because these terms will come at various places. One is known as a transduction, known as foreign DNA induced in a cell by a viral vector or a bacteria vector, and it also describes the introduction of a recombinant viral vector into a target cell. The another term known as a transfection. It is a forced introduction of small molecules such as DNA, RNA into eukaryotic cells, and for introduction of this DNA or a forced introduction of small molecules of such DNA or RNA into eukaryotic cell, certain special techniques are used. These are known as electroporation, means DNA is placed in that particular solution where both the molecules are present, and then pulse of electricity is passed through that solution, and due to this pulses of electricity, the DNA is introduced. This phenomenon is known as a electroporation. There is another technique which is known as a calcium phosphate technique or calcium phosphate precipitation techniques, but I am not going into details of it. But long back, a technique was also introduced to introduce such kind of a DNA into a plant, and that was by a small gun, a pistol-like gun, which was used for this purpose, and that was named as the gene gun. So it is exactly fired near the plant stem, exactly in a similar way we fire the pistol. It consists of small, what you call the structure, which consists of the DNA or RNA solution in it, and if you fire it, just like a bullet, that DNA or RNA will be introduced into the plant stem or wherever we want to introduce it. Another phenomenon is known as a microinjection. This technique is very important and very commonly these days used in vitro fertilization. In vitro fertilization means we are artificially fertilizing an egg in a laboratory with the sperm. In this particular technique which is known as a microinjection, what we do is we actually take a cell, and a small injection needle is required. Very fine needle is there which can go inside the cell. So the DNA is filled in this injection needle, and slowly that injection needle is slowly inserted into a cell which is placed in a small petri dish, okay, and observed from the microscope. So under microscope observation we introduce the needle, which consists of basically the DNA or RNA solution, and ultimately it is introduced inside. So this technique is also known as a microinjection, and overall these techniques are called transfection, or this phenomena is called transfection. Another technique phenomena is called as a transformation, which is basically a DNA uptake by bacterial cell from the environment. So these are the different phenomena which are associated with the DNA manipulation or DNA engineering. But there are certain few things which I would like to tell here, and then when I will record the other lectures, because this phenomena will come at several levels, and these are all phenomena which are basically needed to be understood very clearly. Because everywhere, where you talk about the genetics of a memory, where you talk about epigenetics, whether you talk about the genetics of a color, height, or genetics of a memory, or genetics of a behavior, everywhere the DNA and DNA manipulation is involved. So DNA structure need to be understood very clearly. I already told you that DNA is made up of two strands and made up of nucleotides, which form, so nucleotides form the DNA structure or DNA strands. But this is not only the thing. Actually, the chromosome consists of DNA, or chromosome is made up of a DNA, but outside DNA, to protect the DNA, it consists of certain proteins known as histones. these are two kind of proteins, totally enclosing the DNA structure, and if the histone proteins are not removed, you cannot manipulate the DNA. So before using the DNA strand for a manipulation, you have to remove the histones. In addition to that, DNA, if we see, let us talk about the chromosomes also here. If the chromosomes, as I told you already, that the DNA consists, DNA made the chromosomes. So a chromosome is like a strand-like structure, and there are two strands of a chromosome joined with a chromocenter, which is in present. And in a resting cell, the chromosome consists of a one strand, but when the cell undergo multiplication, whether it is breakdown with a phenomena called a mitosis, in that case the chromosome will divide at the time of division, and from one chromosome another chromosome will form. And if you want to see the earlier structure, clear structure of a chromosome, at the end of chromosome strands, on both the strands, there is a small area at the extreme end known as a telomere. This telomere was recently associated by various scientists that it is involved in the development of a cancer. It is also involved in the development of the aging. It is also related with the senescence and certain other things.